So I'm going to talk about something I haven't spoken about in a long time, and that's herbicide resistance. You picked up on that, huh? You like that one? Yeah. So this has been kind of the topic that is number one. But oftentimes get asked to speak, in like at Ag Week and other presentations, and don't really have enough of a time or an opportunity to kind of do a little bit more of a deep dive into resistance. And so that's what I wanted to do today. And in particular, talk about this metabolic resistance, something that we haven't talked much about, making people aware of it. And so we'll get into some of the different mechanisms of resistance as well. We've been dealing with resistance in this area for quite a while. We've got a whole range of species that are resistant, from the common ragweed, the chickweed in small grains, horseweed or mare's tail resistance, in no-till as well as in small grains. Some of our resistance here is resistant to a single mechanism of action. Some, like the chickweed, some is resistant to multiple modes of action, like the ragweed. It's resistant to glyphosate and the PPO herbicides. We have some populations of ragweed that's resistant to 3 different families of herbicides: PPO, ALS, and glyphosate, which is starting to limit what we can do as far as managing these species. Just because we have resistance and fairly widespread, we can't give up on our resistance management approaches. We're going to talk about some of the biotypes that are resistant, in some cases up to mechanisms of action. Our worst case, I think we're looking at 3. There's still a few products out there that we'll work on. We start losing of important mechanisms of action, we're really going to be in trouble on managing some of this resistance. So what I want to do is kind of review resistance both nationally as well as in our specific region here, discuss how resistance develops, then talk a little bit about different types of resistance, particularly focusing in on metabolic resistance, and then talk about just resistance management in general. The scope of the problem, we have every state has some form of resistance reported, herbicide resistance. There's 75 different species in the U.S. that are resistant to at least one herbicide. So we're not talking about isolated instances. We're talking some pretty widespread problems. It's not unique to any particular chemistry or particular crop. We've got resistance in grain crops, vegetables, fruit, rangeland, aquatic. So no production system, no scenario is really immune from resistance. And by having resistance, what it really does is it limits the options we have for weed control. In the past, there's always been a new mechanism of action coming behind that helped with resistance to help manage it. You kind of think back to the 70s when triazine was first reported, triazine-resistant lamb's quarter and pigweed. And shortly after that, the ALS inhibiting herbicides came out, did an excellent job on those species, worked very well for about 10 years. Then the glyphosate came out, and that worked for about 10 years. And then we had some of these HPPD and PPO herbicides that became much more popular. And in about 10 years, we're starting to see some cracks in them. So the problem is we don't have that next mechanism of action coming along that's going to help us manage this problem. As it relates to our grain crops, the last mechanism of action that was developed was the HPPD herbicides, the group 27s, and that was discovered in the 80s. So we don't have anything that we can replace and kind of plug and chug and keep moving along. Now, FMC has announced last year, I believe, that they have a new mechanism of action in the pipeline. And at the time, I think they said like 2024, 2025, before it might hit the market. I suspect that's kind of an optimistic estimate. There's a lot of hoops that companies need to go through before they register products. So the long and short of it is, we don't have anything on, right in our toolkit that we can plug in and help out with some of this resistance. So we need to maintain the utility of the products that we have. And even though we might have some resistance in our fields and in our area, we still got to be diligent about making sure that they remain viable as long as we can. Glyphosate gets the lion's share of the news when it talks about resistance. This is a slide from Kevin Bradley at the University of Missouri looking at herbicide glyphosate resistance across the U.S. Most of the states have some glyphosate resistance, except for maybe in the upper New England states, maybe some of the western states. But glyphosate resistance is out there. Look at the number of species that we have reported for resistance in the U.S. So it's a widespread problem. It's a very big problem, but it's not the only resistance problems that we have. Before I kind of go much further, I just want to kind of discuss some of the terms that we hear around resistance, make sure we're all kind of on the same board, on the same page on this. First one is like the mode of action. We hear mode of action a lot. And mode of action is that term used for how a plant responds once it's treated with the herbicide. Basically, how does the plant die? We think about amino acid inhibitors. We think about lipids, membrane disruptors. Those are what causes, that's what happens to the plant as it dies. So they're very broad, very large groups lumped together under mode of action. Underneath that, in kind of the hierarchy of how we classify and talk about this stuff, is the mechanism of action or site of action. Those terms are used interchangeably. That is exactly at the molecular level, where does that herbicide interact with the plant? Where is that site that herbicide is binding? What is the mechanism by which it's binding? So it's very specific for that class of herbicides. Within different mechanisms, there might be multiple families, 2 or 3 or maybe even more families. And then within families, there are the active ingredients. And then, of course, a single active ingredient may have multiple trade names depending on how it's marketed out there. But when we talk about the herbicide group number, that number that's on the jugs to identify what what we need to be concerned about from resistance management, that group number is based on the mechanism of action or the site of action. It's not the mode of action, and that's what a lot of times people refer to it, but it's actually that site of action. That's specifically where is that herbicide interacting with the plant at the molecular, the enzyme level to kill the weeds. So to kind of help keep this, sort this out, we developed this chart based on what colleagues in the Midwest have done. It's a herbicide classification chart. We use their design, we use their format, but we modified it for the Mid-Atlantic because, we don't grow sugar beets. They have a lot of sugar beet herbicides in their chart. We grow a lot more vegetables than they did, and vegetable producers also are very much troubled by resistance, so we wanted to make sure we included those herbicides. So we modified it for the region, but this is the chart. It's set up by, first of all, at the high level, the mode of action, how it is that the effect that it has on the plant growth. In this case, we're looking at the lipid synthesis inhibitors. That site of action is the ACCase enzyme. That's where it works. That the group of herbicides that work at that site of action are group 1s. Within that group 1, there's 15 different cases of resistance already reported in the U.S. That's where that 15 number comes in. So within that ACCase inhibitors, there's 3 families: the FOPs, the DIMs, and the DENs. And here are the active ingredients, and here's what the most common trade names are for those different products. That's how we've set up this chart. We also included all the premixes, because this is where it becomes just as important, is understanding the premixes and what are the single active ingredients in those premixes. What are the constituent parts? So a product like Anthem ATZ, for instance, it's got 3 different active ingredients. The pyroxsulfone, which is similar, is essentially it's the Zidua. Flufenicet, which is Cadet, and atrazine. And so we've got a group 15, a group 14, and a group 5 herbicide all within that jug of Anthem ATZ. So we have the most common premixes used in our chart. So I do want to give a thanks to the state soybean boards in the region. They paid for printing this. We did it just about a year ago, and I have copies of it up here on the table. So if you don't have a copy of the chart and would like to get one, just grab one at the end of the talk. So, resistance is not new. We've been dealing with resistance for quite a while. Looking at the number of cases of resistance that we've got here, this chart starts in 1970. Really, I think we go back and look at some of the first cases of resistance were actually in the 50s. When they were first observed, they weren't really described as resistant, but we'll go back to those fields after the whole phenomenon of resistance became better known, and sure enough, some of those were resistant. So resistance has been with us for a long time. Notice that it's really in the about 1990 we saw a really striking increase in the amount of resistance reported in the United States. We get to closer to 2017, 18, and it looks like it might be starting to level off. I guess I would say it's probably not accurate. This information comes from a website by Dr. Ian Heap, thewheatscience.org, and Ian does the best he can of keeping track of this. And what happens is when folks observe resistance, they document it, they contact Ian, he puts it up on his website. But what happens, I think, is it's hard to keep up with that. We've got so much resistance in a lot of areas that what's one more case? So it may not be reported quite as often as it probably should. So I would say that that's probably not starting to level off. It's just lack of effort in some cases of actually reporting them. So I look at this and I don't want folks to think that they might have false hope that, okay, we're starting to get this under wraps, because I think resistance is still spreading. I know resistance is still spreading. It's not unique to any particular chemical class. Like I said earlier, the glyphosate, group 9, really gets the lion's share of attention when we talk about resistance. But if you look at the number of species resistant to any specific site of action, group 2s are the most common out there. The ALS inhibiting herbicides, Pursuit and Osprey and Matrix. There's a big class of chemistry that's there. But group 2s are the most widespread resistance. Group 5, which is the triazines, also quite common. Even in the U.S., we have more cases of the ACCase resistance than we do of glyphosate. So this is not a glyphosate-only problem. But we're talking about resistance. We're not talking about natural tolerance that's out there. We're talking about a change in the weed that that herbicide is no longer effective on. We have a lot of weeds that just are not effectively controlled by certain herbicides. Think of like Select or Clethodim. Spraying on a broadleaf weed has no effect whatsoever. That's not resistance, that's tolerance. That weed has always been tolerant of that herbicide. Herbicide has no effect on that weed. So tolerance is different than resistance. Oops. So you think about morning glories versus some of our Palmer amaranth treated with glyphosate. Pigweed, the morning glory, it doesn't, a lot of times it doesn't die, particularly if it's starting to run, it's got some size. It's not resistant, it's just tolerant to that herbicide. Whereas, there were, for eons, populations of Palmer amaranth that were sensitive to glyphosate, and we could spray it and kill it. However, now we have populations that are resistant to it, and glyphosate has little to no effect on it. If we look at the top 10 species that have reported resistance in the U.S., leading the top is Palmer amaranth and water hemp, followed by kochia, horseweed, and the pigweed complex of redroot and smooth, Italian ryegrass, common ragweed, common lamb's quarter, giant ragweed, common cucumber. Those are the 10 most common resistant biotypes in the U.S. How many of them do we have in the Mid-Atlantic region? Of that 10, 5? At least 9. At least 9. And kochia's in western PA, it's probably in western Maryland, so we probably have all 10 of them in this area. So it's the most common ones are things that we're dealing with here. And, which classes of chemistry are we talking about here, particularly of concern for our area? Now, this is just me kind of sitting down, putting this list together. But I think that probably our top 3 of concern are the group 2 herbicides, those ALS inhibiting herbicides, that big long list. If you think about that chart a couple of them back, it's never a good idea to go back. But that's this group here, this very widespread resistance. That's the group 2 herbicides. That is one that we really have problems with. Maybe not so much in some of our corn and soybean acreage because we shifted to other mechanisms of action, but certainly in our vegetables where we had pursuit, raptor, sandia, that was the backbone of broadleaf weed control in vegetables. Now we've got smooth pigweed that's resistant. We have ragweed that's moving into the area that's resistant to this class. We've got common chickweed that's resistant to herbicides in this class, which includes Harmony Extra. We have Italian ryegrass that's resistant to osprey and powerflex. So this is something that we're dealing with right now without a lot of good options as alternatives. Obviously, the glyphosate resistance, broad, across many different cropping systems. And the PPO resistance. We don't have a lot of PPO resistance. Group 14, that's the Reflex, Valor, Spartan, Kixor, Cadet. We don't have a lot yet, but we do have some. In particular, we have some common ragweed that's resistant to this class. In soybeans, these are kind of been our go-to products for ragweed control. And now, they're no longer effective. And a lot of that ragweed is resistant to glyphosate. So we're losing some options there. So these are probably the top 3 that I have concerns about. Kind of that next group, the group 27s. HPPD inhibiting herbicides, Callisto, Balance, Impact, Loudus, as well as Liberty. Now, I am not aware of any resistance to these two groups in our area. I don't think we have not seen, and no one's reported anything that makes us concerned. However, these are the ones that we have really kind of jumped on to help manage the resistance in this first class. So this is why I put these up here as concerns, because we start to lose activity of these HPPDs or Liberty, we're really going to be in trouble. And then the last kind of class that I have, the triazines, which we've had triazine resistance for a long time. It's just because we've rotated to other herbicides, it hasn't gone away. You know, every now and then somebody, for one reason or another, will go out with just atrazine in a cornfield and right away reminded immediately that, oh yeah, that lamb's quarter and pigweed from 15 years ago is still resistant. So that's out there. The synthetic auxins, and we're going to talk about this a little bit more, but we don't have any in the area that I'm aware of, but with a lot of places in the Mid-South that have switched to this technology for dealing with water hemp and palmer, they're starting to see some resistance show up out there. And then the ACCase is in that list. This is the weeds of concern that I've got. I just want to point out that, remember those top 10 weeds in the U.S.? They're all on this list in our area. So I kind of went through that. I hope, you know, it was just kind of lay the foundation. I hope that was a review for everyone. I hope I don't have to convince anyone that resistance is a problem in this area. That it is, and it's something that as a community that we need to deal with. It's not something that we're going to be able to spray our way out of. You know, we're losing the utility of a lot of these products, and we don't have a lot of alternatives for them. So we really need to put an emphasis on our non-chemical approaches for weed control wherever we can. So you talk about resistance now and kind of go back to some of the basics on resistance. Herbicide resistance is a naturally occurring product or naturally occurring phenomena. Spraying that herbicide on a weed does not cause resistance. That resistance gene was already in those plants before it got sprayed. So herbicides are not changing the genetic makeup of those weeds. Just selecting for those individuals that have the genetic makeup that allows it to tolerate the herbicide. So we spray herbicides and a few individuals survive, and those individuals within a larger population we refer to as biotypes, they survive. They're starting off at a really low population. There's just a few of them out there in the field, and they survive, produce seed, and become problems down the road. They already have that mechanism for resistance in their genetic code. So we're not changing the genetic code by spraying the herbicide. We're selecting for those individuals that have that resistance. So the biotypes, you know, is just a few select individuals within a larger community. Just because a herbicide doesn't perform doesn't necessarily mean that it's resistant. You know, think back 15, 20 years ago. Anytime a herbicide didn't perform, what was the problem? It was the environment, right? Yeah, it was the weather. Now, you spray a herbicide and it doesn't work. Well, it's because it's resistant. Well, neither of those two cases were accurate. And so, you know, just because the herbicide doesn't work doesn't mean that it's resistant. On the other hand, if you can rule out everything else, you know, good conditions, the right spray volume, the right rate, yada, 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 and it still survives, then we need to think about, is it resistance? And the other key thing about resistance is it's inheritable. You can pass it on from one generation to the other. So it's a genetic trait that goes from one generation to the other. So those are some of the traits within resistance. So here's kind of a little slide. You got a single plant out there. It gets sprayed with the herbicide. The blue one's resistant. The others are susceptible. The susceptible dies. That resistant one survives. It grows up, matures, produces seed. Seed goes back into the soil. So then next year, instead of one per field, there's a couple of them out there. So that second year, same thing came back, spray that same herbicide. Everything died except for those few individuals. Now you go from just a few to even more in terms of seed production and contributing to the wheat seed bank, until we get to the point where the predominant biotype in the field is now resistant. I mean, any of these fields where we have resistant weeds, you go out and spray it, you're still going to have some susceptible ones out there. We never really get to that point where it's a complete uniform population of all resistant. There's still going to be some out there that will die, but the majority of them, or at least enough of them of concern, are resistant. There's a great... Should have recorded the first one. There was a point I was going to make right here, and I forgot it. That's what happens at the fourth talk in the morning, Oh well. If it comes back to me, I'll insert it. Okay. So resistance. I know I was going to. I think too. We've become more in tune with resistance as well. And so when herbicides don't perform, and we start to go through our checklist, I think we are much more in tune to resistance now and more likely to investigate whether it's resistance than we were before. So I think in a lot of cases, we're picking up those 10, 15 plants that are at the entrance of the field or in the hedgerows, whereas in the past we may not have thought about resistance and letting it get out of hand. So I think everyone's antenna around resistance is up, and we're much more in tune to it. And I think we're starting to treat it a little bit sooner, which is a good thing. If we can prevent some of these from going to seed early on, we can really help ourselves out down the road. So there's also, some other terms that we use within resistance. We have, single herbicide resistant. It's resistant to only one herbicide. Herbicide A, a specific mode of action, is resistant to only that one. It's a single herbicide resistance. Sometimes, though, we can end up with cross resistance, where we select for resistance by using a single herbicide, and we get resistance to other herbicides with that same mechanism of action. And so a good example of that is the common ragweed that's resistant to Classic. And we had a population that we were working with. We confirmed it as being resistant to Classic. It then treated it with First Rate, Classic, Pursuit, Raptor, Cimarron, Arsenal. No, not Arsenal. Beacon. I don't know. We have Matrix. We treated it with 10 or 12 different herbicides, all within that same family, and they were all resistant with it. So that's a case of cross resistance, where we selected for resistance with a specific herbicide within a mechanism of action or site of action, and it becomes cross resistant to other herbicides in that same group. The other one is multiple resistance. So we select for a weed that becomes resistant to a herbicide. We switch herbicides and start using a different mechanism of action, and we abuse it and overuse it, and now that weed biotype is resistant to two different sites of action. Something that happened early on with some of our common ragweed. We had one of the fields that we were working with, they had used PPO herbicides had repeatedly used them on continuous soybeans, selected for this population that was resistant. They then switched to ALS inhibiting herbicides, used that exclusively, and lo and behold, the population became resistant to both PPOs and ALS inhibiting herbicides. So multiple resistance. And multiple resistance is on the rise. This is, again, website looking at species with multiple resistance. And, you know, there's a lot of them that are resistant to two different sites of action or mechanisms of action. But look here, you know, he's got a couple of them that are at 9, 10 different modes of action. The same weed resistant to 11 different mechanisms of action. And so I didn't realize we had that many different out there, but it's a scary problem when you start getting your options become very, very limited at that point. So, say all that kind of a background on resistance, but you know, I've talked about resistance, and what I want to kind of do now is shift a little bit and kind of look at a little bit more nuance in terms of different mechanisms of resistance, because we have different mechanisms conferring resistance in the certainly across the U.S. and I want to talk about those individually. So we have altered site resistance. We have resistance of overexpression of the target enzyme, compartmentalize or sequestration of the herbicide, and then want to talk a little bit more in depth about enhanced resistance or metabolic resistance. These first two, altered site enzyme and overexpression of the target, is dealing with right at the enzyme where that herbicide interacts with it to cause death to the plant. These tend to be very high levels of resistance, where you know, we can spray. Think back to like the atrazine resistant with the lamb's quarter and pigweed. You know, it'd spray ungodly amounts of herbicide on those plants, and you saw little to no effect. You know, on the other hand, we've got some resistance where it might only be 2- or 3-fold differences. So there's a whole wide range of resistance that we see out there. So we can go from a 1x difference in activity up to well over 1,000. And to some extent, if we know that level of resistance, we can kind of determine what mechanisms it might be, particularly with those first two, the altered enzymes and the overexpression of the enzymes. We get very high levels of resistance. So the target site enzyme, high level resistance, we show little to no response of the herbicide. You know, this was a mutation of the target protein. Herbicides can't bind to it. This is kind of the classic example. You know, it was the first ones reported back in the 70s and 80s, and happened with the atrazine and the triazines, as well as the ALS inhibiting herbicides. So this is kind of a simple diagram of how this happens. So you have an enzyme in the plant, you have a substrate, they come together, produce a product that the plant needs to survive, and the plant goes on just merrily. All right? But when you use a herbicide comes in, and it attaches to that enzyme, and because the herbicide is attached to it, the substrate cannot fit in. It's not, that enzyme's not able to make the next product. The plant suffers, the plant dies. So that's how these herbicides often act. What happens with a target site mutation is that place where the herbicide normally binds with the enzyme is altered, so that herbicide can't attach to the enzyme. Because the herbicide can't attach, the substrate comes in, binds with the enzyme, makes the product that it needs, and the plant just survives. So this is kind of the classic example of target site enzyme. And this is the one where we generally have a very high level of resistance. This is an example of that from our greenhouse with some common ragweed that's resistant to ALS inhibiting herbicides. We sprayed Pursuit on this common ragweed. Now, we know Pursuit's not a very good common ragweed herbicide, but when you spray 100 times the normal use rate, you'd expect to see at least some effect on common ragweed. But no, this is a really high level of resistance, up to 250 times. Now we're starting to look at maybe a third reduction in biomass. This is an altered enzyme, gives us a really high level of resistance. Also, we sprayed these plants with, in terms of cross resistance, these were the ones that we sprayed with Sandia, which is very good on ragweed, Classic, Harmony. This is the one we had about 10 different individual active ingredients, and it was we basically saw the same results with all those products. So target enzyme that conferred cross resistance to numerous herbicides within that family. The other group that gives us high level resistance is its gene amplification. Again, we'd show little to no effect from the herbicide. And what happens is that target enzyme within the cell, there's an overproduction of it. So there's not enough herbicide to bind with all the enzymes that are there. The only case that we are aware of this at this point is with glyphosate. So this is what typically happens in the cell. You get these little enzymes, the ESPS enzyme, the ones that look like the little packet that binds with it, substrate, the amino acids that the plant needs, and it dies. So that's kind of what happens in a very simplistic way. But when you get gene amplification, you just get so many more of those enzymes produced. Notice all the glyphosate is still a enzyme, but there's just so many enzymes there that the substrate's still able to bind, still able to produce the amino acids. The weeds just go on just happily without any ill effect. So that's what happens with gene amplification. The other mechanism of resistance is a non-target site. So this does not affect the enzyme itself, but it's other sites within the plant. And these tend to be fairly low levels of resistance. The plants will show some type of response. The weeds survive by limiting how much herbicide actually gets to the enzyme. This is what, here's a little diagram. This little thing here is the herbicide. It's supposed to get into the cell and bind with the enzymes that are in the cell, but it's not able to get across the cell membrane at the concentration it needs to kill the plant. So this is something we see with glyphosate. You spray glyphosate on resistant mare's tail, and you see a little bit of stunting, you see a little bit of yellowing. 2 or 3 days after you spray it, you go and you look at it, and you have some hope that it might die. You go back a week later, and it looks just it grows out of it. So we'll see. Like mare's tail, spray glyphosate on it at our normal use rate. We might see 40, maybe a 50% reduction in growth, which isn't enough, but we do see some response. I talk about when we first reported glyphosate-resistant mare's tail. We did a couple things in the greenhouse, and it said, yes, this is the type of research we need to explore. We had this huge experiment. We had 3 or 4 different collections of mare's tail. We sprayed 2 or 3 different formulations of glyphosate on it. We had multiple rates. It was a huge project in the greenhouse. I went in there like 2 days after I sprayed it, and just about every plant that was sprayed all looked the same, whether it was susceptible or resistant. And I thought, well, there's a couple days work down the drain. But a week later, I'll go back, and those that were resistant turned green, started growing. Those that were susceptible died. So with this type of resistance, we do see some effect on the plant. Another mechanism of a reduced translocation is rapid necrosis, where the herbicide acts so fast that the plant just shuts down and the herbicide doesn't get distributed where it needs to, and eventually the plant recovers. This is a case with glyphosate-resistant giant ragweed, where they've seen this. They spray ragweed on the susceptible plants versus the resistant plants. The susceptible plants just turn brown and die back almost immediately, whereas the susceptible plants start to turn that glyphosate yellow and chlorosis and eventually die. But what happens with the resistant ones is kind of shut down so quickly, glyphosate doesn't move to the cells where it needs to, and the plant survives. Reduced translocation, that's what we see often with these PPO resistance. This is some common ragweed. These two down here are the susceptible, untreated, a 10x rate of reflex. These two up here are the resistant, untreated, a 10x rate. So we're getting quite a bit of leaf burn and stunting with these higher rates of reflex, but the plant recovers and they're still quite competitive. And in terms of cross resistance, here's the response that we see with Cobra, Valor, and Resource, other PPO herbicides. A lot of leaf burn, but we're not killing them. So reduced translocation from causing some level of response in the plant. And the last one I wanted to talk about was metabolic resistance. And this is something that's kind of come to the forefront. This is something that we're just in the last year and a half, there's been some reports, some very disturbing reports about resistance using this mechanism. It's generally not discussed as much because it wasn't very widespread, but weeds start to degrade herbicides or metabolize herbicides the same way as crops do. So, you think about spraying, 2,4-D on wheat. Well, why doesn't that wheat start to show any effects? Because the herbicide is metabolized. Dual on corn. Dual we use for all these other grasses. Why doesn't it kill corn? Because corn is able to metabolize it and convert it into an inactive compound very quickly. So this is happening now. We're starting to document this happening with weeds as well. Some of the more recent reports are these group 15 herbicides. That's the duals and the acetochlor, Zidua falls in that group. Resistance reported in Kansas, this is Arkansas, to resistance of palmer amaranth to these group 15s. Water hemp in Illinois, resistant to group 15s. In Kansas, they reported resistance of palmer amaranth to 2,4-D and dicamba, all by this type of this mechanism. So we're starting to hear more of this. Like I said, it's very similar to how crops avoid injury. It typically is a kind of a three-phase approach where the herbicide molecule has a slight modification to it. It's then often joined with another compound, often a sugar, and then the cell kind of says thank you but no thank you and just kind of shuttles it off into a place where it's not going to have an impact. So here's kind of a diagram of it. This is the cytoplasm of the cell. The first step is fairly minor. The herbicide just loses a couple of carbons and hydrogen atoms, but that allows for a sugar to bind at that site. And so now the herbicide is bound with the sugar, and the cell then sends it off to a vacuole or to intracellular space where it's just has no effect. So this is metabolic resistance that we're seeing. It involves this whole class of enzymes called cytochrome P450s. These enzymes facilitate a large number of metabolic processes, usually detoxifying compounds that normally would injure the plant. It's a really large number of genes that are involved in cytochrome P450. You know, rice has been documented to have over 360 genes involved with cytochrome P450, you know, but humans only have less than 60 genes for cytochrome P450. So plants have a lot of these compounds in them to help detoxify something that could harm it. And herbicides are just kind of falling into that category. You know, we know that this is a very powerful way of detoxifying herbicides because there's a number of herbicides on the market that have safeners that accentuate the activity of cytochrome P450 and increase crop safety. You think about dual 2. Well, that 2 on dual has a compound in it that speeds up the activity of cytochrome P450, makes corn a lot safer, or makes dual a lot safer to corn. We see that with status. There's safeners in status. The Q products of Accent Q, Steadfast Q, and so on, that's got safeners in there. So we're using that to our advantage to increase the crop safety. But these P450s are important in detoxifying toxins to the weeds, including herbicides. So this is that water hemp from Illinois. 2 different populations of water hemp versus 2 susceptible populations. They sprayed a number of these group 15 herbicides. This just happens to be S-metolachlor or dual. This is the one they saw the most effect with, or the most response to. And these 2 populations behaved very, very differently when they were treated with S-metolachlor. Now, you know, you might look at these rates and say, well, those are extremely low rates. Well, remember, this was done in the greenhouse under ideal conditions. And so while these may be low rates, these low rates were killing the susceptible populations, whereas the resistant one was not. So, you know, don't just kind of look at the rates and kind of disregard this, because the key point here is that these populations are behaving very, very differently. To a product like a long-chain fatty acid, which we for a long time thought was not going to be as big of an issue for resistance. So this is group 15s on tall water hemp. This is 2,4-D resistant and palmeramran from Kansas. So the top is their resistant population. This is their susceptible population. Sprayed it with a range of rates of 2,4-D. And while even at some low rates, we're starting to, they see some response. It's still surviving up to a 16x rate. So, and this is due to enhanced metabolism within the palmeramran. So by developing metabolic resistance, it confers the concern is it's going to confer resistance to other chemical classes that have never been treated. So because these are such broad acting enzymes, that by selecting for those that are resistant to, say, break down 2,4-D, they may also be breaking down other herbicides that have never been applied. In fact, with this population was also resistant to mesotrione or to Callisto because of metabolic resistance. And while they haven't proved it, their suspicion is it's being this 2,4-D is being detoxified the same way as mesotrione is being detoxified. So selecting for one resistance, could be selecting for multiple resistance and not even know it. And because this is such a large group of enzymes, we said over 600 genes involved, it's hard to predict which genes are involved in metabolizing these herbicides. You know, we've got the chart up here on modes of action and can predict modes of action and where it's acting. And kind of the reverse side of this with metabolic resistance, we can't necessarily predict that by rotating certain classes of chemistry that we're not selecting for metabolic resistance. We could be. That's the concern. And it's very difficult to predict because of so many different families. Done? Okay. All right. And a lot of genetic diversity within the weeds. All right. So how do we handle it? Would we handle this any differently than we do other types of resistance? No. I mean, again, this is not a situation where we're going to be able to spray our way out of. We're going to have to rely on non-chemical strategies as much as we can so that we retain the utility of the herbicides that are there. But because we're starting to see, maybe not in our area, but in the Midwest, where they're starting to see some of this metabolic resistance, it has the potential to move to this area as well. So rotating and using non-chemicals as much as possible. And you know, where do you get some of this information? We've myself and a number of colleagues in the region and across the U.S. have developed this herbicide resistance website, Grow IWM, which has a lot of good resources on integrated weed management, chemical strategies, cultural strategies, mechanical prevention, and so on. So growiwm.org is the website. We're remodeling and retooling this, so in a couple of months it's going to look a little bit different, but there's a lot of good information up there now. We're going to continue to put up information for the foreseeable future. So, time for a question or two.